Well, good morning. Man, if I hear the phrase path of totality one more time, I'm going to have a stroke. I'm so sick of that.
Get on that path.
We're not in the path of totality. Okay, so we are kind of in the middle, not really the middle, toward the end of Roman numeral 9. So recapping quickly before we pick up. So we started with a whole muscle, removed the skin of someone's arm. Follow that arrow down here. That's the entire muscle. Have the outermost boundary, not shown here, but it's in your notes, the fascia. The steep fat is the epimysium. Hold, they bind and hold together the subunit. What are those subunits? Also contribute to the tendon. And so then we go to the next level. We're looking at a fascicle. Each fascicle has an outer layer called a perimysium, which binds and holds together the subunits, which are muscle cells. Go to the cell, and we have the outer boundary of the endomysium. Deep to that, the sarcolemma, which is the cell membrane. They bind and hold together the myofibrils. The subunits of the myofibrils are actin and myosin, collectively known as myofilaments. This picture of a single cell, they've pulled out one myofibril. We can see the filaments. We have the area right, just making sure you can see the cursor, and you can. We have the area right here that is just myosin. We have the darkest area right here where actin and myosin overlap. We have the lightest area down here, which is just actin. In this very dark area where actin and myosin overlap is what gives us this striated appearance, which is there really aren't any vertical striations. That's the area where actin and myosin overlap. So as we pick up with this, names have been assigned to these different areas. In other words, the area that myosin, etc., so that you can talk about it. It's kind of like in the cell cycle. People got sick of saying the time during the cell cycle when the double-stranded chromosomes split at the centromere and each sister chromatid, now a single-stranded chromosome, moves toward an opposite pole. Let's call that anaphase. Then all we have to say is anaphase. We don't have to say all that other stuff. So the same thing happens here. So if we start with that area, that line, that's a Z line. And we don't see the other Z line. It would be roughly down here. We'll see it in a minute in a different picture. But these Z lines, plural, are the boundaries of a sarcomere. So all of that sarcoterminology, sarcoplasm, sarcolemma, sarcoplasmic reticulum, comes from the term sarcomere. The sarcomeres are the functional units of a muscle, just as the alveoli are the functional units of the lungs. The nephrons are the functional units of the kidney. The sarcomeres are the contractile units of a muscle. So we know that muscles contract, they shorten, and they typically move bones. They do other things, smiling, for example. But when a sarcomere contracts, it's going to become shorter. These filaments are going to slide across each other like this. As far as naming these different regions, I'm going to start with the simplest of pictures. So that's a Z line. That's a Z line. They are the boundaries of the sarcomere. Now, in this picture, you can see we actually have two sarcomeres, the one on the left and the one on the right. And we have this area that's just actin. And that's the I band. So the I band is just actin. You can label the I band as I just did at the end of a sarcomere, or you can let the I band span two sarcomeres, as they had already done in the labeled picture. The point of the I band is it's just actin. We're looking at the I band on the right of this sarcomere and the I band on the left of this sarcomere. Collectively, they are an I band. They have a Z line running down the middle. But we could just as well label the I bands like this. Doesn't matter. It's the area that's just actin. It does or does not have a Z line running down the middle. Then we have part of the sarcomere. Look at that closely. It runs from one end of myosin to the other. That's called the A band. Which myofilament or myofilaments do you see in the A band? You see both, correct, actin and myosin. Now, if we break the A band down, we have the part of the A band where actin and myosin overlap versus the part of the A band that is just myosin. The whole thing is A. Remember, A runs from one end of myosin to the other. But as we look at that myosin myofilament, we see that in the center of the A band, there's an area that's just myosin, and on each side, an area where actin and myosin overlap. The H zone is the part of the A band. So within the A band, just myosin. Now we've seen repeatedly things that don't make sense, like when they call the fossa on the proximal end of the tibia condyles. And you know, we just have to picture the elders messing with us. So they give all these regions a letter, but guess what they call these two areas where actin and myosin overlap? The area of overlap. It's extremely descriptive, but there are a lot more letters left in the alphabet. Why didn't they pick one? Just dumb. So, area of overlap. So that is within the A band. Where actin and myosin overlap. I often call the area of overlap the AOO. That's not some standardized abbreviation. I don't think you'd see it in any book, or if you Googled AOO, I have no idea what you would come up with. But it's just shorter for me to write than area of overlap. Now remember, the sarcomeres are the functional units, the contractile units. The sarcomeres shorten. Actin and myosin slide on each other. Again, like this. We can see that if we compare the top picture to the middle picture. Look at the, if these two Z lines have moved in, then the I band is narrower. The H zone is narrower. The question becomes, how do the filaments slide? But let's first write down how the bands change when the sarcomeres contract. To just try to maintain context here. So this will be Roman numeral 10, changes in bands upon contraction. Get it large as I can, but yet you can still see the labels up there. So again, Roman numeral 10, changes in bands upon contraction. So what happens to the I band? Look at it here, look at it here, and look at it here. So the Z line basically slides right up against myosin. So it gets much narrower. Now remember, the A band, look at your notes if you have to, runs from one end of myosin to the other. Oh, God. So here's A labeled up here, one end of myosin to the other. Is A any different in the middle picture or the lower picture? No, it's unchanged. Because A runs from one end of myosin to the other, unless myosin got shorter, A couldn't change. A band is unchanged. All right, as we look at the H zone, remember H is the area within the A band that is just myosin. If you compare this picture to this picture, what's happening to the H zone? Huh? Oh, well, it gets, I mean, a little bit of it there. Don't look at the bottom yet. We have a special word for that. So the H zone narrows and overlaps, yes. So the exact numbers you come up with here are irrelevant. But what is the distance between the two marker caps? A couple of feet, whatever. Like I say, the exact number doesn't matter. Now what's the distance? Speak, people. Now what's the distance? Now what's the distance? 0. Now what's the distance? Negative. Negative. The H zone narrows and becomes negative.
What's that vertical line?
It's the M line, and we'll get to that. But I'll go ahead and label it for you. It's another protein called myomycin. The consequences of those actin filaments overlapping. So remember, you use a stain to dye a tissue. In this case, the stain binds the protein. So the more protein there is, the darker the darker it is. So we have this area where we have actin and myosin overlapping, that is the darkest. What's going to happen in terms of darkness, lightness upon complete shortening, complete contraction of the sarcomere? That area is going to get what? Even darker. start here where you can see the labels. So get your bearings. We have 2 Z lines. So that's a sarcomere from here to here. Get the lights, please. The very light areas on each end of the sarcomere, those are the I bands, and you can see the label up there if you need it. And then myosin runs from here to here. So that's the A band. And these 2 parts of the A band, why are they so dark? That's the area of overlap. And then within the A band, we have this lighter area, which is the H zone. And Pablo, this dark thing we see right here is the M line. Now, if you look at the M line, it's got a kink in it. That's just a tear. When you work with tissues, imagine taking a sheet of paper and slicing it into 1,000 sheets. Think how thin that would be. So tears just occur. Now, as this sarcomere shortens, look at how the H zone is getting narrower. Because these actin filaments are moving closer together. Look at how the I bands are changing. As the Z lines are moving toward the end of myosin. And so if the 2 actin myofilaments actually overlap, as we see here, what's going to happen to the H zone? Yes, it becomes negative, but in terms of darkness, lightness, what's it going to do? Let this guy in. Good morning, Christopher. Okay, so the H zone is going to get darker. Look at that. Back away from it to see it. So it's using pictures like this that allow us to understand what happens and create diagrams like this. Imagine it had to be figured out. When a sarcomere shortens, how does it shorten? Maybe the filaments coil. Think about DNA. When you go from discrete chromosomes that you can count to chromatin. Chromatin to chromosomes involves the DNA coiling. The coils being maintained with proteins called histones. So it gets shorter and thicker. DNA literally goes from long, skinny, thread-like, invisible to short and thick. And we can see the discrete chromosomes. For all we do, that's what happened here. But it's not. The filaments don't coil, they slide. So then the question is, how do they slide? How do they slide? Well, it turns out, and I'll give you a lot more magnification in a minute, but it turns out, what's the blue myofilament? Thank you, I had forgotten. Myosin has these little heads that dangle from it. And those little heads are going like this, pulling on actin. Lights, please. This is actin. This is myosin. My hand is the head. My wrist is the neck. So the myosin head is going to bind actin, swivel, release, bind, swivel, release, bind, swivel. That's how that's what's pulling the actin filaments in. So we need a lot more magnification to see that. So let's get it. So let's start up here and get our bearings. So we've got a sarcomere from Z to Z. The red are actin. The blue are myosin. So we have this area on the end that's just actin. What band or zone is that? I. Then we have the area that goes from one end of myosin to the other. What band or zone is that? A. And within A, we have the area that's just myosin. What's that? H. And within the A band, we have the two areas where actin and myosin overlap. What are they called? AOO, areas of overlap. Notice that what we see here, we haven't seen before, are these little, dear God, that got big. These little bud-like things hanging off of myosin. Those are the myosin heads that I spoke of. So if we really blow it up, follow that arrow. So this is a myosin myofilament. You can see the little heads protruding. So we have the tail fibers, which is what all of this is minus the heads. Then we have the part that swivels, called the neck or the hinge. And then we have the head. So those heads, look up here, those heads truly bind actin, bind, swivel, release. Bind, swivel, release. Now here's actin. Don't worry about the orange versus the yellow. So the orangish, yellowish, spherical things constitute actin. And where on actin do these heads bind? They bind what's called the active site, also called the binding site. So the heads on myosin bind the binding site on actin, bind, swivel, release. Bind, swivel, release. All of them. Now the binding and swiveling has to be in perfect synchronization. Kind of like if we were on a big boat in the ocean and it was windy. And there's 50 people on one side of the boat and 50 people, and there's no engine in the boat. It's an old boat. There's 50 people on one side of the boat and 50 people on the other side of the boat. And we're rowing the boat into the wind in perfect synchronization. What if every one of us put our oar in the water, pulled, and then took our oar out of the water? What if that's how we were synchronized? What would happen when we all take our oar out of the water? We would move backwards. So the perfect synchronization is that each one of us must be 180 degrees out of phase with the other. So if I'm behind Jocelyn, when I put my oar in the water and pull, she has her oar out of the water and is moving it forward. So these heads all binding, swiveling, releasing in perfect synchronization. They're not all binding and pulling at the same moment. If they were, then when they all release, the sarcomere would elongate. Wild stuff, huh? Now, as I look at actin, and I think about the fact that the heads on myosin bind the binding site. Again, binding site, active site, same thing. It looks to me like the binding sites are blocked. They're blocked by this green thing, whatever that is. What is that? Oh, that's a protein called tropomyosin. And I see another protein connected to tropomyosin, these little globular things. That's another protein, troponin. If you work in a clinical environment, you've heard of troponin because troponin is a cardiac marker. If someone's had a heart attack, not chest pain, a real heart attack, a myocardial infarction, death of heart muscle tissue, their troponin will be elevated, and you see that in the blood. So troponin and tropomyosin are regulatory proteins. Recapping a little bit of this. The myosin myofilaments contain these heads blown up here. They bind the binding site on actin. They swivel. The binding is called a cross bridge. And when they pull or when they swivel, it's called a power stroke. So the myosin heads bind the binding site on actin, and then they swivel, or they undergo a power stroke. But the problem, again, is that I don't see how this head can bind the binding site because tropomyosin is in the way. So let's do that thing that we've all heard of, but I've actually never done. You start a new book, a good book. It's a murder mystery. No, I've never read a murder mystery book. That's not what I'm into. But we'll pretend it is. And about halfway through the first chapter, you're just like, oh my God, I've got to know who did it. So you skip chapters 2 through 15 and you read the very last chapter. Find out who did it. We've all heard of that. I've never done it. Don't know if you've ever done it. But let's do that here. And we'll do it with a very simple picture. Now, first of all, people taking a photograph of this, fix this. And if I wasn't the way I am, I'd grab the box and stand up there and fix it. But this is a myosin head. It's not a cross bridge. It's a cross bridge when the head binds actin. Publisher knows about the mistake, but the author is dead. The book is out of print, so they're not going to fix it. Like all A&P books, there are two authors. There's an anatomist and there's a physiologist, because nobody's really both. There's no such thing as a degree in anatomy and physiology. So Kent Vandegraff, he's the anatomist. Stuart Fox is the physiologist, and Kent's dead. Stuart's still alive. But nevertheless, if you're photographing this, and you should be, cross that out and change it to myosin head. But we see the same thing here. We see the head. We see the binding site on actin. We understand that the myosin head wants to, if we personify it anyway, it wants to bind that binding site, but it can't because tropomyosin is in the way. It's blocking the binding site. And we see troponin is connected to tropomyosin. So just like we saw here, we see tropomyosin spiraling around actin, blocking the binding sites, and we see troponin is connected to tropomyosin. As we jump to the last page of the last chapter, because we're dying to know how we get that binding site exposed, notice that there's a little thing on troponin. It turns out that's a binding site for calcium. So look at the next picture. Calcium has attached to troponin. Troponin and tropomyosin have moved to the side, allowing the head to bind actin. That again. So there it is at rest. The binding site on actin is blocked. See this little thing on troponin, it turns out that's a binding site for calcium. And here we see that calcium has bound troponin. Troponin and tropomyosin have moved to the side. And the head has not only bound, but look at the angle through the neck. So the head has bound and undergone a power stroke. Again, look at the angle through the neck there. Look at it here. So as soon as that head binds actin, it sounds just like that. Listen to, get up close here. Contract your biceps for me. I heard it. It's what it sounds like. So how in the world does all this happen? Where does the calcium come from? Well, you'll probably be sorry you asked, because I'm going to tell you. I'm going to tell you with a series of pictures, and I suggest you photograph each one. So the pictures are numbered. We don't care about the verbiage. We care about the picture. This is what you want a picture of. Now, after we go through this, I'll recap it writing the steps on the board. There'll be 10, 12, 15 steps. The reason the number of steps is not absolute is because the number of steps are not real. We make up the steps to tell the story, to give you something to wrap your brain around. It's like if you were telling me about a movie. You wouldn't say, well, step 1, he parked the car. Step 2, he got out of the car. Step 3, somebody hit him over the head and stole his billfold. You'd tell a story, like a paragraph. So, but after we simply look at the pictures and I talk about it, and you're recording me, and hopefully that thing is recording me. That it is. Been for 41 minutes. Oh God, I didn't know we had been here that long. All right, so here we're at rest. Notice that the myosin heads are sitting here. Notice that the green thing, tropomyosin, is spiraling around actin, the orange and yellow thing. And the yellow parts of actin are the binding site for these myosin heads. Just looking again, making sure you can see the cursor. We know from a previous picture that to expose that head, calcium binds troponin right here. And then troponin and tropomyosin move out of the way. The head binds actin, and a power stroke occurs. You can kind of see both together. We see calcium in the picture. And interestingly, we notice that the heads, not all of them, but a couple of them, have an ADP and inorganic phosphate attached. I wonder why that is. Find out. Now, in picture number 2, we see that calcium has attached to troponin. And as soon as calcium attaches to troponin, that pulls tropomyosin out of the way. Look, the binding sites are all exposed now, the binding sites being the yellow portions. As soon as those binding sites are exposed, look at that. The myosin heads have bound the binding sites on actin. What's that called when the heads bind actin? Cross bridge. Now look, those heads are still holding an ADP and a phosphate. We don't even know where it came from. We go back to picture number 1. When we started and everything was resting, the binding sites on actin are blocked. The myosin head has ADP and phosphate connected to it. Wonder why? Where'd it come from? Then we looked at picture number 2. Calcium has attached to troponin. That pulls tropomyosin off of the binding site. And then in picture number 3, the heads have bound the binding site, forming cross bridges. In picture number 4, we see the heads releasing the ADP and the phosphate. Also look at the angle through the neck. There's picture 3. The heads have just bound actin. cross bridges are formed, but the heads still have the ADP and the phosphate. As we go to picture number 4, look, the ADP and the phosphate's being released. Look at the angle of the neck change. This is almost a linear, almost a straight line right here through the neck. In other words, up here. Versus after the, what's it called when it power stroke. Now we've got a right angle here through the neck. And again, we see the ADP and the phosphate being released. In picture number 5, it looks that the heads are binding another molecule of ATP, and the cross bridges are broken. In other words, this head is going to bind that molecule of ATP, but the head is no longer connected to actin. So the cross bridge in this case is broken. Same with the one down here. So apparently, the myosin heads have to bind ATP to break those cross bridges. If there's no ATP, the cross bridges can't break. This is the basis of rigor mortis. When you die, your ATP is depleted, your muscles remain contracted. You've all seen that dead animal on the side of the road all contorted. And after death, time of death, the rigor begins to set in, reaches a maximum, and then decay begins. Decay. It's like the roast that you left on the kitchen counter. You forgot it, went away for spring break. You came home 10 days later. So when decay sets in, the rigor begins to subside. And so the coroner can estimate the time of death by the state of the rigor. And is the rigor increasing? Has it maxed out, or is it decreasing? Wanted to know that. Wanted to give you the visual of that roast on the counter. Could have used a dead cat on the counter, but that would be even grosser. Now you have that visual too. So the heads bind ATP, the cross bridges break. Where'd the ATP go? Here the head is binding ATP. Cross bridge is breaking. Now the head swings back up. What's it done to the ATP from here to here? It split it, yeah. And so now the head, the sarcomere is resting again. Calcium's still attached to troponin. I still see I still see calcium attached to troponin. I still see all of these binding sites on actin. And if you compare, let's see, I think we've got them all here. If you compare this picture, this is where we started. The head is not attached to actin. It's holding ADP and phosphate. We wondered where that ADP and phosphate came from. Well, now we understand that that ADP and phosphate here that we started with are there because to break the cross bridge, the head had to bind ATP. As soon as it binds that ATP, the cross bridge breaks and the head splits the ATP, the ADP, and the inorganic phosphate. It's not wild. And imagine in one muscle, millions of sarcomeres, each containing millions of myosin heads, bind, power stroke, bind, that's a cross bridge, power stroke, release, bind, power stroke, release, over and over and over. All right, let's this into some steps for you. About the time here, but I'll give it a shot. Okay, so this is for context. Roman numeral 12, molecular events of contraction. The sarcomere is at rest. Myosin is blocking the binding sites on actin. See that in the picture. The green thing is tropomyosin, blocking these yellow parts of actin, which are the binding sites. The head is holding an ADP and inorganic phosphate from a previously split ATP. Now recall, and you've told me previously, what makes muscles contract? They don't just contract because they want to. Motor nerves. So the nervous system, motor nerves, neurons, a single neuron innervates a small group of muscle fibers. What do we call that? Two words, motor unit. So now what do we call the signals within neurons? It's an electrochemical signal. It's similar to electricity. What are those signals called? Action potentials. So we have an action potential in a motor neuron. So this goes back to this picture that we've used repeatedly. My elbow, biceps brachii. Muscles innervated by a particular nerve. Now there's a space So I've called it a nerve here. That's a gross structure. Remember, the nerve is divided into many nerve cells. The muscle is divided into many muscle cells. A single nerve cell or neuron innervates a small group of muscle cells or muscle fibers, and we call that a motor unit. So here I'm showing a single neuron. It's going to stimulate a group of muscle fibers. So that's a motor unit. But notice there's a space, physical space between the end of the neuron and the muscle fibers. Some of you know what this space is from Psych 101, called a synapse. Now, because this synapse is between a motor neuron and a group of muscle fibers, it has another name. Myoneural junction. And it takes special, thank you, Jocelyn. It takes special chemicals called neurotransmitter substances to move the signal across the synapse. So this is abstract enough, but try to appreciate that when I say action potential, all you know about it right now is it is similar to electricity. So the motor neuron zaps the muscle and the muscle contracts. But recall that we will get to the point of talking in detail about action potentials. Take us 30 or 40 minutes. And then we'll talk about synaptic potentials. That's a synapse. Then the synaptic potential is moving the signal across the synapse. And what did I say? It requires special chemicals called, there you go. The most common neurotransmitter in the body is acetylcholine. So we'll talk about that one for sure. The other one we'll talk about to give us a clinical application is serotonin. Because a lot of people have not enough serotonin and they suffer from clinical depression. They take drugs like Paxil, Zoloft, Zyban, Wellbutrin. Talk about some of those. So we're sitting here at rest. The heads have an ADP and a phosphate attached from previously split ATP. We get the action potential in the motor neuron. Then we're going to have the release of acetylcholine at the myoneural junction. Release of acetylcholine at the myoneural junction. Camera back on the main board. Sorry, Christopher and Baxter. I wonder if they're hanging out together. Baxter may have invited Christopher over. Baxter spent the weekend with, well, so did Poppy, with their mother. They both came home sick. I think she does that on purpose. She overfeeds them stuff that, like bean burritos. That's good for a dog. And then she asked me, I wonder why they pooped in my apartment. You shouldn't feed them bean burritos. Okay, where are we? Number 4. The action potential spreads across the sarcolemma. What's the sarcolemma? Cell membrane, okay. And into transverse tubules. Sarcolemma spreads across the cell membrane and into the transverse tubules. And we're still sitting there at rest. Picture number 1. Where's that calcium come from? Know that when calcium is comes into play, there it is, it binds troponin and the binding site's exposed. But where does the frickin' calcium even come from? So the action potential spreads across the cell membrane and into the transverse tubules. The sarcoplasmic reticulum releases calcium. Calcium binds troponin. Finally, we get to change pictures. Troponin pulls tropomyosin off of the binding site. And that's where we are specifically in this picture. Calcium is attached to troponin. Troponin has pulled tropomyosin off of the binding site. We see all of the yellow parts of actin exposed. Of course, we know what's going to happen next. The head's going to bind actin. Actin heads bind actin. This forms a cross bridge. So we have the head attached to actin. That is the cross bridge. The myosin head releases the ADP and inorganic phosphate. And this drives the power stroke. On the next picture, we'll see the calcium being released. The angle through the neck changes. Power stroke has occurred. Binding, power stroke, releasing, cycle, continues as long as the binding site is exposed. As long as calcium is attached to troponin, which means as long as action potentials are coming in, calcium will remain attached to troponin. Binding sites exposed, this bind power stroke release will happen over and over and over. The head must bind molecule at the end of each cycle, bind, power stroke, release, bind, power stroke, release. The head must bind a molecule of, say, of what? The head must bind a molecule of ATP at the end of each cycle to break the cross bridge. So when your 3-year-old says, "Mommy, Daddy, how do muscles work?" What you're going to tell them? Get out the whiteboard. Dare I ask questions on this? Any book you might have bought, remember I told you, you need a book. Doesn't matter what book. A lot of you bought a $20, $30, $40 book. Good. But any book you have is going to show this series of pictures. May not be identical to these, but they'll be close enough to make the point. But also Google and then find it that way. Please understand, this is physiology, not anatomy. Memorizing those frickin' terms on that terminology worksheet seems like child's play. Use the animations on D2L. Last module, oh, maybe it's the last module anyway, it's the online resource module. Either in the video submodule or the muscle submodule, you'll find animations that show this happening. The coolest thing in the world. Watch those, and you probably have to download them to your device to watch them. Different people are going to have different experiences with them, especially depending on whether you're using a Mac or a PC. But you're intelligent creatures, you can figure that out. All right, so we're going to pick up with Roman numeral 13, because clearly ATP is required. This is an energy requiring process, so we'll start just by summarizing the roles of ATP in muscle contraction. And I'm shutting down the recording.